Thank you all for being here. It's a really good crowd, and it's nice to see so many familiar faces, but also some new ones, so welcome. So I'm going to go ahead and get started. As Jamie said, I'm getting my joint degree in the medical school and a PhD in E-IPER, and what that means is I'm fundamentally curious and concerned about diseases that affect humans and how social and ecological systems affect infectious diseases. So one thing to note is that infectious diseases are the result of interacting epidemiological, environmental, and social behavioral systems. And these are complex systems that require interdisciplinary approaches to understand. Many people ask me when I talk about my work about Outbreak or Contagion, the movie. Many of you have seen that. There's well, when is the next big epidemic going to occur? And we cannot predict with precision when and where this will happen. But I argue that we can better prevent and respond to these events if we understand the conditions in which these rare events emerge. So most emerging infectious diseases originate from non-human wild animals, the vast majority. And the first event, or initial event, of an animal infection going to human is considered a zoonotic spillover. And then when an infection can transmit from a human into other humans, we consider that an animal or a human infection. An example of this is simian immunodeficiency virus, which transformed into HIV, which people are familiar, has had a huge impact on human societies. So while we cannot predict, we do know that particular areas around the world have increased risk. And we've identified those areas with higher risk, which are noted in yellow, because they have these are forested tropical areas, and they have a high mammalian diversity and are undergoing land use changes. And we can talk a bit more about why these factors might coalesce to form an environment in which infectious diseases can emerge. So I'm going to take you through a framework that I created in order to test hypotheses on the ground for how different layers of the system interact and allow for the emergence of an epidemic. So the first layer, I'm thinking about landscape, and in tan is human-modified land, in green is forested habitat. And so in this conceptualization, I imagine that humans persist or live primarily in human-modified lands. Animals are primarily in forested habitats. And the way that the environment is organized provides a context in which they can have contact and interact. The next step in this framework is thinking about how this positioning of animals and humans across the landscape and the contacts, whether there are certain demographic features in human behaviors that might influence whether or not there's an event which exposes a human to infected bodily materials from an animal. After that, you think about the spillover of an infectious disease, and that is often the result of multiple or repeated or risky exposures to an animal infection. And lastly, we can use other methods in thinking about if this infection can get into a human population, what is the context in which, and what are the social relationships that might drive that infection disseminating through a population. So I'm going to test hypotheses at each of these stages. And so this is a roadmap for the rest of the talk, going through the hypotheses, the first being the forest loss and habitat fragmentation increase the likelihood of human-primate contact. The second one being specific subsistence behaviors, that's a tongue twister, and demographic factors predict levels of exposure to primate infections. Higher levels of exposure to these primate reservoirs of disease increase the likelihood of a spillover event. And social structure is affected by social affiliations, and it can contribute to epidemic potential in these sparsely populated areas. And then we'll talk a bit about how these factors interact. So I'm going to take you to the field site. As I showed you a map before, there are certain areas that have a context in which we anticipate there being a higher likelihood of infectious disease emergence. I have circled in red Uganda, which is where we're going to go now. Kibale National Forest is located in the western part of the country, near the border of the Democratic Republic of the Congo. Kibale National Forest was established in 1993, at which point people who were living in the park were forced out, and the border of the park is maintained by the Uganda Wildlife Authority, so it's a protected area. Reasons why this specific site might be conducive to emerging infectious disease are there's a very high level of biodiversity, over 220 species of trees and other other aspects of biodiversity. There's a high abundance and diversity of primates, one of the highest richnesses of primate groups in sub-Saharan Africa. There's a high prevalence and diversity of pathogens from these primate populations. There's increasing rural population density. It has twice the average rural population density of the rest of the country, with 240 people per square kilometer. It's undergoing continuous deforestation. Much of the land has been converted into agricultural plots. There's human encroachment on these animal habitats, and there's wild animal encroachment on human habitats. So looking back at the framework, I'm going to try and understand how these landscape patterns and configurations influence when and where people have contact with primates. So the hypothesis, again, is forest loss and habitat fragmentation increase the likelihood of human-primate contact. And one thing to note is many models of landscape change and interspecies contact rely on the assumption that these host populations vary with the proportion of land converted. And so this idea of land conversion incorporates ideas of fragmentation. So if you start with a fully intact forest ecosystem, there's a boundary between human-modified lands and forested habitat. As you begin that fragmentation pattern and kind of deforesting that area, you get an increased border between human-modified land and forest. And ultimately, you get these forest fragments, which are small islands of forested habitat that are embedded in human-modified landscapes. And what happens within these fragments often is that there's a higher density of primates, there's more opportunities for infection transmission, and potentially greater borders in which humans and primates can move between landscape types. And I refer to this as edge density, or the amount of border between human habitat and primate habitat, and use that as a metric, or use that to interrogate whether or not these changes can lead to increased contact. So with that in mind, all these processes happen at a landscape level, but the way people move through space and potentially interact with primates happens at a very local level. And so trying to understand what the landscape features are around a home is something that I thought would be a unique contribution, thinking about how landscape actually affects people at their households. And so what I did is created a 300-meter buffer, which has been cited in the literature in this particular area as being the area in which people perform subsistence behaviors and access forest resources, and looked at fragmentation and deforestation at that scale. So I collected surveys in the site from people who had had contact and people who had not had contact. And one thing to note is that I have households in which no one has had a contact event, and households in which at least one individual has had a contact event with a primate. Collected data on GPS locations of these households, and GPS locations of actually where the contact events occurred, to try and understand, is it not only where people are situated in space with their household, but also, is it the specific type of landscape type in which people are interacting with primates? Is there other features of that that can give us insight into why these events might happen? So I classified high-resolution imagery, 6.5 meter resolution, to look at how much deforestation and land change had happened between 2011 and 2015. And I bounded this period because I collected my data in 2014, and because there was reliable cloud-free imagery from this time period. What we found is that there was 7% total forest area deforested during this time. But the really important part of this analysis is that 46% of this deforestation was happening in the fragments. And so these unprotected islands of forest habitat, which still contain primates. And I excluded fragments that were less than 0.8 hectares and were more than 100 meters from the nearest other forested habitat, which is what primatologists have noted are the criteria for presence of a primate within those habitat types. As you can see, there's a wide variation in the amount of deforestation that's happening at a very localized level. And so you can imagine that the effects of deforestation and edge density, being the amount of boundary between human habitat and forested habitat, is quite different across the landscape. So it might have potentially different impacts or effects on the activities or the potential for contact between humans that live in that specific area. And what we found is there were significant differences between the amount of deforestation and the edge density between households that had had a contact event and those who had not. And so you can see in red, or the orange color, are contact households, and in blue are the households that had no experience of a contact within them. We also looked at the areas around the specific contact points where people and animals had actually had a physical direct contact event. And what we found is that the edge density was even higher in these areas. So potentially in very fragmented landscapes, you're getting even higher potential for contact with a primate. However, there was only 57 of these confirmed locations. It's very hard to get people to take you exactly to where they had contact, and there's probably some variation in that. But as you can see from this data, there are major, the contact locations have a higher edge density and deforestation than even the contact households. So thinking about these factors as predictors for whether or not a household would have the experience of a contact event, or an individual within that household would have the experience of a contact event, we found that edge density was the most significant predictor, and deforestation was also a significant predictor. What was surprising was that there was no significant relationship between household distance to a forested area, which has been cited in the literature as being a significant contributor. But the odds of having at least one contact increases with each unit of edge density. And the unit of edge density is meters over meters squared. And what this means is that there's minimal increases with each unit of edge density, but at very highly fragmented landscapes, that probability may increase quite a bit. And you saw within the first slide that there is a range of 14 to 256 in the amount of edge density that exists around these households. And then the odds of having at least one contact increases with each unit of forest loss as well. So what we found from the study is that landscape pattern and deforestation localized around a household does seem to affect whether or not an individual is likely to have contact. This provides some empirical data for theoretical models that have been presented in the literature, but it also points to a greater need for understanding the behaviors at the very local level. We had this bounded area, which we assumed that people were having contact in, but really understanding the behaviors and maybe the inner household differences, demographic factors, and the behaviors that people are engaged in might give us even more insight into what's driving contact between humans and primates. So moving on to the second part of the framework, which is looking at how human behaviors and demographics may influence both contact, but also the level of exposure. Because when we're thinking about the possibility of infection going into a human from a wild animal source, you think about repeated contacts or the force of that contact event, the intensity and possibility for bodily fluid exchange. So the hypothesis is that specific subsistence behaviors and demographic factors influence levels of exposure to primate infections. And the literature has focused primarily on bushmeat hunting as a subsistence behavior associated with increased risk of primate contact and primate infection exposure, because it's a very intensive activity which requires cutting of meat and potential for blood-to-blood transfer. However, there are potentially other behaviors, particularly in a context like Kibale National Forest where bushmeat hunting is not customary, that are leading people into contact. We know people are having contact with primates, and it's not all random. There's some behaviors that might be leading people into contact with primates. And so we conducted a behavioral survey on forest use and human-modified land use to try and understand what features exactly about the individuals might be predisposing them to have contact. So what we found is there was 11 different species of primates that were involved in these contact events. There were 64 of these individuals were having what we consider fairly minimal contact, which is touching a dead primate, which doesn't seem to pose a huge risk of infection. 18% of people in the study slaughtered a primate, even though bushmeat hunting is not part of their culture. 3% were attacked by a primate. The most notable case was a woman who lost her arm to a chimpanzee when she was a young child. So there are cases and examples of these really intensive contacts that you anticipate might lead to the sharing of bodily fluids. So in thinking about the level of exposure and what are the demographic factors, we looked for individuals who had multiple contact events or repeated events, and then individuals who had intensive contact events. Oops, sorry, that should be with contact. So what we see is there's a difference in sexes, with males having higher levels of contact. However, we wanted to understand whether it was the sex or those demographic characteristics that were providing the impetus for why contact would exist, or or was it a specific behavior in and of itself that was leading people into a context in which they might have contact? And so while food foraging, or looking for food sources within the forest, pole collection, and poles are small trees which people use for building fences or housing, and forest agriculture were all activities that 16 to 17% of the population were engaging in. However, they had very high rates of contact, or people that were engaging in them were having higher levels of contact than other activities. And as you can see, that's also skewed primarily towards males engaging in these activities. So trying to parse apart, is it the fact that they're male, or are there specific qualities about the demographic, or is it the behavior in and of itself that's putting them in this position to have contact with the primate? So these are the kind of things we were trying to get at in this study. And one thing we found is that 14% of people within these intensive contacts were guarding crops. This has been cited in the literature multiple times, that guarding crops in this area has been a big problem and an issue for food security. Many children are not going to school because they're staying home, particularly male children, to guard crops. So we know that there is fear of primate crop raiding, and that it's apparently leading to these more intensive contacts as well, when people are actually killing a primate. And what we found in logistic regression, and I apologize for the resolution of that, but it's that being male, when you put all the factors, both demographic and behaviors, into a model, being male was the most significant, along with doing agriculture on your domestic lands actually reduced your risk of contact. And so that is not parsing apart whether or not it's the demographics or the behaviors, but when I created a conditional logistic regression model, which matched by sex, matched by age, matched by community, I found that forest activities such as pole collection, forest agriculture, and foraging for food were very significant, irrespective of whether or not the individual was a male or a female. And actually, those activities were not only significant for predicting a single contact, but multiple contacts and contacts that lead to increased exposure. So we found in this study that specific forest activities are predictive of contact, repeated contact, and higher intensity contact events. Males are more likely to have contact, perhaps facilitated by these gender roles within these communities. And it points to a greater need for an understanding of how much these activities are related to infection risk. Because we see potentially a risk of having these contact events, but trying to understand whether or not zoonotic spillover is an actual risk is the next area that we are interested in when we're thinking about zoonotic emergence. So then thinking about the incidence of spillover, or whether that spillover is happening and how related it is to exposures. We ask and hypothesize that higher levels of exposure to primate reservoirs of retroviral infections increases the likelihood of spillover. So you anticipate people who have blood-to-blood contact or potential higher exposure are potentially more likely to be having these retroviral infections. And the underlying assumption is that people who have intensive or repeated contacts are at higher risk. And we've seen that from the literature in individuals who are bushmeat hunters or are having repeated contact events with primate reservoirs of infection. So I collected blood at the household and processed this into serum-packed blood cells, and it was also preserved on FTA cards. And one thing to note is, though the power went out pretty much every day, we were able to keep the cold chain intact, so we feel confident that our results are accurately presenting the infection level within these communities. Use beta-actin to confirm the integrity of the DNA. And one thing to note is that we felt that there was a potential for infection from primate retroviruses, because primate retroviral infection is quite high, in the high 90s for simian foamy-virus. And for red colobus monkeys, they have a 10% prevalence of simian T-cell lymphotrophic virus and primate T-cell lymphotrophic virus. And there's also been recent research on emerging hemorrhagic viruses in a number of these primate groups. And so you would think in that context, where people are having contact with primates, there's some potential for there to be a spillover event. So I screened these samples at the lab of Bill Swetzer, who studies primate viruses using a number of techniques to screen for known viruses that have been identified in primate groups. And then for samples that were positive, or we had a high suspicion that there was a high level of human-primate contact or potential for spillover of novel or for bodily fluids to be shared, we deep sequenced those at the lab of Tony Goldberg at the University of Wisconsin to look for whether there's anything emerging, not only viruses that we know about, but potentially other viruses. And what we found was that there was a very low incidence of infection of the individuals we sampled. 1% were infected with simian foamy-virus, which, to note, does not cause, from all evidence, does not cause disease in people. It has only been used as a marker potentially for primate-human contact or the potential for bodily fluid to be shared between a primate and a human. In this population was sex-balanced. Just as many men were infected as there were women. And only 5 of the individuals who were infected with simian foamy-virus had had primate contact, or individuals reported they had primate contact, leading us to question what the route of exposure is and how people are actually getting that infection. There's not been great published literature on other routes of infection, potentially through feces or urine in the environment, but that hasn't been well documented. Less than 1% of individuals who we screened were infected with human T-cell lymphotrophic virus, which originates from primate T-cell lymphotrophic virus. And this population also was sex-balanced, and only 1 individual had had a primate contact event. And human T-cell lymphotrophic virus can be transmitted human to human. This contact event was with the red-tailed guinin, which in Cameroon, individuals who had HTLV, actually HTLV3, which there's 4 strains of HTLV, had contact with guinins. So that is a potential exposure, but we're not putting any money on that bet that is where that came from. But basically, the story is that we can't identify the mechanism of infection. And so there's still a lot of questions about how people are getting exposed to these primate viruses. So the contributions of this study were that we added to the known number of people that are infected with primate retroviruses. We know there's a pathway that spillover can occur because we're finding primate retroviruses in human populations. But primate contact in this setting seems to be posing very minimal risk of infection. We're not finding very many cases. And perhaps it is not a huge priority for these communities, even though I think as a public health community and as people who are interested in emerging infectious diseases, we think of it as a very important area. It may not actually be a high risk in this place. And some of what we found when we combine what we discovered in this study with what was discovered in the previous study is that there's these exposure events or behaviors and landscape features that may drive people to have contact with primates, which we assume is the precursor to exposure, but that there are no associated predictors with the infection. So where do we focus our efforts? So spillover primate retroviruses is low. Does this mean that there's a risk of emerging infections or epidemics? And so this is a bit of a rhetorical question because of course there's still a risk and there's still avenues for infections to spill over. But we could not identify any particular mode of transmission because much of the infected population did not indicate a primate contact event. However, what many of you probably know is that one spillover event can have very devastating consequences. And therefore, we can't discount the fact that there have been infections that have gone from primates into humans in this context. So if we want to understand how the social and behavioral and epidemiologic characteristics may relate to the epidemic potential, we can start to interrogate how social relationships dictate how infected populations could come into contact with susceptible populations. And one way we can do that is by using simulation techniques. We didn't come into a context and find the outbreak of an epidemic or an infectious disease, but we can kind of understand how it might transmit through a population. So moving into the last part of the framework, thinking about zoonotic spillover and how that may lead to a local outbreak or an epidemic, we can use social network techniques to understand whether a zoonotic infection can become an epidemic. So in this last section, we're going to think about how social structures may facilitate infection transmission and ways that individuals form relationships and can drive that. So the last hypothesis that we'll focus on today is that social structure is affected by social affiliations, and this contributes to epidemic potential in these sparsely populated areas. And one thing to note is that in these communities, people are living in quite far from one another. Even though I said it was a high-density rural area, it's still a rural area, and you're not commonly coming into contact with people unintentionally. It's usually through venues like churches or markets, or people in your household, or people that you choose to spend time with, such as close friends. And so egocentric data provides us a representation of the entire network and allows us to investigate the ways in which individual connections can contribute to relationship structures across an entire network. So since you can't sample the entire population, you can get insight into how certain individuals form friendships with their close friends or form relationships within their community. And you can use attributes of an individual or affiliations of an individual to understand whether friendships are formed based on similarities or differences in factors that are associated with an individual. So is an individual friends with people who are similar in age or very different than age? Is an individual friends with people who are at the same church or market, or people that are at very different churches or markets? And so we can use these attributes and affiliations to describe the similarities and differences in friendships. So this is one technique that we can use to interrogate and estimate what covariates or what factors generate relationship structures is egocentric random graph models. Exponential random graph models. I always say that, sorry. And what these do is they allow you to predict ties that do not exist or that you don't know about in the network because you aren't interviewing everyone within your network. And so what you do is you use the variates or the covariates that you find out through your egocentric network data collection, and you can predict ties between individuals based on these variables. So if you know an individual forms friendships with someone who's similar in age, and there's other individuals in the network that fit these same characteristics, you can predict the potential of a tie between those individuals. And with that, you can form an entire network. So infection simulation is one technique that we can use when we don't have an infection of interest that we're tracking. Because if you don't have an infection that you're looking at the epidemiology of, you can kind of understand how it might move through a population. And so in using simulations, you can look at how a susceptible individual is exposed to infection, becomes an infected individual, and depending on where they are in the network, may have very different effects on an epidemic. So when you simulate an epidemic in these modeled networks, you don't have any say over where that infection goes. And so it could go into an individual pair or friend group pair that's very separate from the network, or they could be very central to the network. And so you can see that just stochastically, you can get very different epidemic effects from seeding an infection within a network. In our particular communities, we found that the quality or the characteristic that was most predictive of community structure and why people were choosing, or how friendships were formed, was community affiliation. And that's intuitive in the sense that in these very sparsely populated areas, people are most likely to form friendships with people who they're living close to. These areas are not paved, there's not much mode of transportation, and so clustering around communities seems intuitive. However, that might be frustrating, it lacks a bit of precision, but it's what the data tells us at this point. So once you're able to model multiple structural properties, or you create a model from the data, and then you're able to fit that model to the data. And what we found is that based on our network data that we collected, when we were able to generate a network based on community structure, the distribution or the number of ties within the network fit quite well. And so the last thing I want to note on this, since I'm running out of time, is that these structures matter for whether or not an epidemic can be transmitted within a community and how far that epidemic may reach. And so what we did is we simulated hundreds of networks based on the data, using community as a factor that facilitated friendships or clustering of people within a landscape. And when we seeded an infection randomly into that population, we got very different infection dynamics than when you had a network in which the ties were randomly distributed and were not distributed based on what community a person was part of. And what that tells us is that in this setting, community affiliation or social structure contributes to the potential infection dynamics in a simulated network that's modeled on a real-world network. Community affiliation in this context is the most significant factor that's driving friendship formation. And that even in sparse rural networks, there's potential for zoonotic spillover to result in an epidemic that can reach high prevalence. So, in conclusion, we went through the many different layers of this framework. We talked about how landscape pattern may influence contact between people and wild animals, specifically primates. We talked about how behaviors and demographics may influence the quality or the intensity of those contact events. We were able to investigate whether zoonotic spillover was present in highly exposed populations, and whether or not in a simulated network or representation of the network that we observe in these places, the spillover of an infectious disease could emerge and become an outbreak or an epidemic with far-reaching potential. And so just to return to the original theory about these interacting complex systems and infectious diseases that are at the center of them, we know that each of these factors can contribute to whether or not we see an epidemic or the emergence of an infection from wild animals into human populations. And in Uganda, this is particularly important. There's a lot of development underway. Many of the roads are being paved. These very rural areas are very quickly being connected to very urban centers where the emergence of a retrovirus or any other infection from a wild animal source into a human population could have even wider reaching and disseminated effects. It's the second fastest growing country in sub-Saharan Africa. There's a high population density moving into many of these rural areas. And in these places, many of these vulnerable forest fragments, which host many populations of primates, are quickly being decimated. So there's a lot of transition and a lot of dynamics happening, which I think will affect all the different outputs that we saw from this framework. So it's really important to take some risks when you're doing research and to go out there and investigate things that you don't know how they're going to turn out. I think one really important point I want to leave you with is how essential it is to return to communities that you work in these very vulnerable settings. When I worked with individuals in this context, I returned results from the infection data that we took. Many of them said I was the first researcher who had come back and given them individualized results, but I feel like it's our ethical imperative as researchers to do that. And as an individual who's experienced zoonoses in my own life, which led to a very devastating event as a young child, I know these events are not constrained to these very rural sub-Saharan areas. They are globally distributed and are things that I don't think that we can turn away from and we need to turn our attention to, even if we don't have evidence that they're occurring currently. And with that, I want to thank everyone. It took a village, or like an entire team of people, to make this research happen. Many funders, my family and friends, and many research collaborators to make this work happen. And I just want to thank you all for listening and look forward to questions.
I think we'll open it up to questions. This is a kind of multidisciplinary thesis that is so timely as you look at the news today that Ebola has just spilled over to a major city in the DRC. So we look forward to future work from Dr. Bloomfield. Okay, let's open it up to questions. Yeah. Yes.
You mentioned a childhood story in the end?
Yes.
Is that something you're interested in telling?
I'm happy to tell it. I don't know if this is the right crowd or context, but I was the recipient of 0157H7, which is an emergent hemorrhagic E. coli variant. And so I was hospitalized for over a month and was very aware of the interconnections between humans, the environment, and animals. Maybe not consciously, completely as a young child, but started to think about that and think that is a major reason why I am curious in this type of work. Yeah. Yes?
I was wondering if you could speculate on, for this case in Uganda, do you feel like it's like a critical case? Like this is the place you'd be most likely to see contact or spillover? Or is this sort of like the average anywhere where you have forested landscape that has primates in it or some other case?
I think it's representative of a landscape that's very biodiverse, that's undergoing a lot of change, landscape and population density included. I think, as we can see from the retroviral infection data, it doesn't seem to be a prime example of places we're seeing a lot of spillover. I would think Cameroon or the Democratic Republic of the Congo, where bushmeat hunting is much more customary. We are more likely to identify infections. I still think it's valuable, and I think it's really important to understand why people who are not targeting primates as a food source are still coming into high contact with them. It seems to be driven primarily by habitat overlap and kind of this development process. And so I do think it's important not to ignore that. However, it's just one point in a long trajectory of landscape change and transition. Unfortunately, this is just cross-sectional data, and it would be really valuable to actually have dynamics of the system, so at very different time points and seeing how different stages of deforestation or human population growth were actually affecting those properties and those outputs. But yeah, I think it's a great example because I work there, but I think there's probably ones you would find higher incidence and spillover. Yeah. Yes, Alan.
Thanks for the great talk. I love the photos, especially, as well as the scientific content.
You can like the photos more. That's it.
I was just wondering, you mentioned that you did viral metagenomics, but you didn't present any of that work. Did you end up doing that? Is there anything interesting to share?
No, I mean, TTV, so Tinovirus, what is that? Tinovirus was the only thing that we found that we were able to deep sequence. I don't work in genomics. I was working primarily with the postdoc in Tony Goldberg's lab. So in general, we didn't find anything that was that interesting or that wasn't completely prevalent among all individuals. I would like to know more about the HTLV strain that was untypable, but at this point, wasn't able to generate any more information on that. But yeah, I'm curious to learn more about that. That's kind of like the next frontier, but I didn't do a lot on that. Yes, Gerald?
I was wondering for your serological analysis, because you did a DNA-based PCR and DNA assays, does that assume that retrovirus is that there's a productive activity? So if you were to go back and look at RNA, would that capture more exposure?
Yes, you possibly could. Yeah, it assumed that there would be more replication. So yeah, no, that's a great idea. I didn't have the time to go back and do it again, but I would love to. I do think we might find more infections if we did that. Yeah.
Something you and I talked about years ago, I'm wondering what your perspective on this is now, is what is the consequences or implications of your work on primate conservation and conservation efforts? If we know that they pose a risk for spillover that may actually undermine conservation efforts of wildlife, and so how do you kind of balance that philosophically?
No, I think that's a great question. Of course, if you kill off all the primate populations, you have no risk of retroviruses. But I think there's this intermediary stage where a recent publication I had with some collaborators is this intermediate stage of deforestation has the most likelihood of having infectious disease spillover. So I think that is one thing to think about, right? Like what part of the transition are you in? You can't just decimate an area without thinking there'll be any consequences. When I interviewed people locally, people on average viewed primates as pests and caused a burden on their livelihoods and particularly food security. But we're really concerned about maintaining forests and forest habitat. So I think there are better ways to orchestrate community engagement in the conservation process. Not knowing enough about how that would work, I do think that there's value in figuring out how to engage communities in a real way on how to maintain these primate habitats. I think the story that the forest fragments are the most vulnerable tells us that conservation efforts have been really successful in maintaining the boundary of the park, and it's really these areas that are unprotected that have both the highest risk of contact and the highest risk of being lost, like the populations of primates as well as the landscape. So I think there's some balance between those two, but I think maintaining the protected areas is definitely an important one. I want the monkeys to stay. I don't know, or the primates, sorry. Primates, not monkeys. But I don't really know how to address that question, unfortunately.
Yes. I thought your network analysis was really interesting. And graphically just really cool to look at, but also that you had this really interesting result that came out of it, that it's not network density, but it's actually transmission would go faster through communities. And just as someone that doesn't know what is currently being done, how might that change the way people actually try to deal with with an epidemic if one did break out, like knowing that it's through communities?
Yeah, it really depends on the infection that's occurring. So, right, for something like flu, you would target vaccination interventions in a particular way. So if you notice that it was happening in one community, you might set up a vaccine like health fair or something of that nature. I think the problem with having community as the resolution is that like it's frustratingly like diffuse, which means I think community captures some of these other meeting areas such as churches or markets, and there's just too much missing data for them to be valuable predictors of the community structure. So I think it actually warrants more investigation of what are the factors actually driving those connectivities, because I do think it's a product of the type of data and the way that we modeled it. I think it's true, but I'm just not sure how resolved that is. I think, depending on the infection, you could have these vaccines, or you could, I worry about like targeted campaigns sometimes because you're targeting specific people or types of people. And so you'd have to think really ethically and consciously about how you were going to do that. But yeah, I definitely think there's ways to do it. That's a great question. Yeah. Yes.
You mentioned sort of dynamics at that end.
Yeah.
And this is like a, I guess, impossible question. But
Great.
thinking about the other,
Love impossible questions.
thinking about the other major anthropogenically driven environmental change that we all think about a lot here, how would climate change like fit into this framework, both in terms of influencing the ecological system or affecting the human system?
Yeah, so Uganda's at the equator, so it's a pretty steady temperature, but there have already been climate effects. Rain patterns have changed. That would definitely change where fruiting trees are within the population or within the landscape. And they found that when fruiting trees, which are affected by rainfall, are decreased within the forest, primates are crop raiding more frequently. And so there's definitely going to be like dynamics of at least where agents are moving in space that are highly affected by climate change. I'd be really curious to think about how that actually affects the way people are moving through space or some of these other activities that are driving the different layers of the system. Yes.
I'd like to ask you a question about gender balance.
Yes.
And the fact that males were so much more likely to have exposure, but yet in the simian, I know there are low numbers of infected people, but in your virus infection, it was very sex balanced.
Yeah.
Was there an activity that predicted that women would?
Yeah, so poll collection, even though very few women did it, the women who did engage in it were highly likely to have contact with a primate, and as well as forest food foraging. So those are the two that were most highly predictive, irrespective of gender, for precipitating a contact event. And one thing that I was talking about with a research assistant yesterday was, what is it about poll collection that might be driving this contact event or high intensity contact events? And one thing is when individuals go in for poll collection, you're often carrying a machete because you need a machete to cut down a tree. And so there might be these factors such as like just having a machete in hand, being attacked or being approached by a monkey that leads you to having a more intensive contact event. But that's completely speculation. I would have to look into that more.
Yes.
So what are you kind of thinking about as next kind of future directions, either for your own work or for this area of research? What are you excited about?
Yeah, I mean, I think one thing I'm really curious about is the longitudinal nature of many of these changes. We see such dramatic loss of the forest fragments over even 4 years. There was a 46% reduction that I think having another set of data to compare this to and say, it was the edge density, or is it the change in edge density, or is it this other dynamic that's really driving the system? Because I think even though we tried to kind of control for the fact that we had this cross-sectional data by looking at landscape in parcels and not as a whole, I think a lot of the dynamics might change when you think about time passing and what happens, particularly with friendships, the social structure that exists, and how that may be changing with this increasing development. So I wouldn't do the same thing again, of course. I'm like, just do it over again. But I would think about like how to add a time dimension and how important that is potentially to driving some of these things. And also, I mean, I think the climate change effects and how that might be influencing these differences in movements and where agents are in space is something I'm really curious about. Yes?
How do you see that translate to like a physical bat vector and non-human mammalian pathway?
Sorry, a what?
Like for another vector, like a bat, for example, which how would you see that kind of fit into, or how transferable would that be with this primate-focused analysis?
Yeah, so bats are highly abundant in this landscape as well, and I think there have been 3 Ebola outbreaks in Uganda in regions not far from Kibale within the past 15 years. And so people are quite aware and attuned to not only the fact that they probably shouldn't touch primates, but specifically they shouldn't be touching bats. So I think the contact would definitely be significantly lower. But I think doing an analysis of people going to fruiting trees or other areas where there could be bat droppings that carry infection you could do a similar type of analysis as people are foraging for foods, which we know is an activity that leads to primate contact. It might be an activity that also leads to contact with animal feces that carry infection. So I think there's a lot of room for that. There's actually some PhD students at Tony Goldberg's lab and the University of Wisconsin who are looking for emerging bat viruses. And so there's been a number of at least preliminary studies or preliminary evidence on the fact that there are new coronaviruses that are coming out of bat populations within this area. And that is a particular concern because as an infection that's much more easily transmitted, that might be something that would have a higher impact on people and people being able to pass that on. Yeah.
How market dependent is your sample population, and also do they have market dependent, like subsistence versus ownership?
Yes, sorry.
And do they have livestock?
So about 80% of people indicated that they were subsistence farmers. The amount of livestock per household varies widely. Cows are extremely costly, and so very few households have many cattle, but there are a number of chickens, and a number of goats that households have. But I would say it's very subsistence based, and people do aggregate at markets, but as we saw, that doesn't seem to be like a driver of at least the social structure, and probably not a lot of the economics is based on that. But there are, of course.
Or just to put your spillover data in context, what's the largest example in sub-Saharan Africa or in Saharan Africa of spillover? Is there, how large does the percentage get?
Of how many people are affected by spillover? Yeah. Well, the Ebola outbreak in West Africa is one prime example where over 11,000 people died and 28,000 people were hospitalized from what is anticipated to be one spillover event.
Okay. And is there any research that's been done as to how many contacts compared to the amount of spillover?
No.
In that
I don't think so. I mean, there's been evidence from bushmeat hunting populations on spillover of simian foamy virus, which is something I talked about that doesn't have consequences for human health. And what they found in many of those studies is that people who had more intensive contact, such as bushmeat hunters or like an injury from a monkey or a primate, had 24% prevalence of simian foamy virus, whereas individuals who had very minimal contact had about 1%, and the individuals who were kind of in the middle had like 12%. So it seems to map quite closely with like repeated contacts or level of contact, at least for retroviral infections. I can't say what it would be for other types of infections. I left a lot of time for questions. I didn't realize I was talking so quickly, but I can keep answering. All right, you can go. Okay.